Hello everyone, welcome to our seminar. Today's topic is long-term live cell imaging with Luminicell Tracker cell labeling kit. Before we start, let me briefly introduce our company. Our company is called Luminicell. It was founded in 2014 by Professor Liu Bing from National University of Singapore. And in 2022, Luminicell became a wholly owned subsidiary of Nanolumi, which is also a Singapore-based company. As the parent company, Nanolumi is an advanced luminescent materials company, and it has 3 different verticals focusing on different areas of applications. And Luminicell is the vertical focusing on advanced bioimaging. Why do we need long-term bioimaging? We know that in biomedical research, and especially the end-to-end translational research, there is a need to study the occurrence and progression of the disease, and also other biological processes. And we know that all those processes take time. For example, if you are a cancer researcher, you may want to study the occurrence of a primary tumor and also the whole process of metastasis. Imagine if you are a stem cell researcher, you may want to track the distribution of the transplanted stem cells and also the fate and viability of the stem cells in the long term. And a similar problem also exists in other biomedical research fields, including the immunotherapy and organoid, spheroid research, tissue engineering, and also others. So here comes the problem. How can we study what we can't see? Fluorescent labeling is the most commonly used labeling method. And here are the currently available fluorescent labels. But remember, all these technologies fall short. Fluorescent proteins, such as GFP and RFP, are commonly used for long-term tracking. But the preparation is laborious, and it takes weeks of preparation in advance. And furthermore, fluorescent proteins are invasive. It requires a researcher to manipulate the genetics of the cells. Therefore, the technology is not suitable for the cases that need non-transgenic methods. The common organic dyes, especially the common small molecular organic dyes, including the cyanine-based dyes and fluorescein or rhodamine-based dyes, they are prone to photobleaching, meaning the signal is easily weakened. Therefore, the common organic dyes are not suitable for long-term tracking. And the third type, quantum dots. We know that quantum dots won Nobel Prize of Chemistry last year, and they are considered as one of the brightest materials in the market. But the metal cores of quantum dots exhibit toxicity to cells. They can cause the cells want to track to die. Therefore, quantum dots are not the best choice for live cell imaging. To meet all the need of long-term bioimaging, Luminicell proposed a new solution. Briefly, Luminicell trackers are nanoparticle-based fluorescent trackers. And in the nanoparticle, we have 3 different layers. You can also imagine as 3 layers of innovations. Let me introduce from the innermost layer to the outermost layer. So here, in the core, we have encapsulated the AIE fluorescence dyes in the innermost layer. AIE is short for aggregation-induced emission, and is a phenomenon first discovered by Professor Benzhong Tang from Hong Kong University of Science and Technology in 2001. So what's the specialty of AIE dyes? Here, let me show you the differences between common dyes versus AIE dyes. For common small molecular dyes, they have a planar structure and they are highly emissive in solution, but they loses the signal in aggregates due to pipette stacking. While for AIE dyes, on the contrary, the typical AIE molecules are non-emissive in solution, but they become highly emissive in aggregates due to the restriction of intramolecular motion. And here is the emission change of ACQ and AIE dyes when adding different ratio of anti-solvent. And here, the video clearly shows the emission change of ACQ and AIE dyes upon adding different ratio of water. We can see the emission is weakened for ACQ dyes while the AIE dyes got emission intensified. In the core of nanoparticle, the dyes are always in the aggregate state. But we know the ACQ dyes will lose signal in aggregate state. Therefore, they are not appropriate to be the core materials of the fluorescent nanoparticles. Well, on the contrary, AIE dyes perform the best at the aggregate state. That's the reason why we encapsulate the AIE molecules as the core materials in our nanoparticle. Now we move on to the second layer, nanoparticle encapsulation. Here we have a patented platform to encapsulate AIE fluorescent dyes with amphiphilic lipid polymer matrix into nanoparticles. And our platform can help us produce the nanoparticles in a reproducible and uniform way. And in addition, the nanoparticle size can be tuned between 20 to 90 nanometers. Our encapsulation technology platform enables the challenging materials and hydrophobic materials to be biocompatible, so that they could be used for in vivo applications. Moving on, let's talk about the third layer, surface functionalization. Beyond the nanoparticle encapsulation, we can further functionalize the surface of nanoparticle with different targeting and functional groups. We have our own proprietary methods for conjugation of different biomolecules, including the aptamers, peptides, and also antibodies, so that we can control the cellular uptake efficiency, and we can also enable the selectivity towards tumors or towards a specific organ. You can see the sophisticated design here. This is to integrate all the necessary benefits into one system to satisfy the needs of bioimaging. With all this design, we have our products here. These are the products we have been selling for years. Luminicell Tracker 540 and 670, which are emissive in green and red respectively. We have both cellular labeling kit version and also vascular labeling kit version. For cellular tracker, the products have been optimized with the best cell uptake, allowing a wide variety of cell tracking applications. And for vascular labeling kit, the products have least cell uptake, also meaning there is no leakage of the fluorescent nanoparticles from the blood vessels. We have been distributed by Merck Millipore globally since 2017. And we have also established the distribution partnerships with companies in different countries or regions. You can purchase the products through the distributors or directly with us. Beyond those mature products, we have also released new products. We have expanded our spectra. Now we have the blue product that can meet the multiplexing needs. Moreover, we also have the NIR1 and even NIR2 products for applications that require deeper penetration depth to redefine the imaging limit. We foresee the increasing importance of NIR and more applications in the near future. And these are the vascular labeling version. This page shows us the absorption and emission spectra of all our 5 products. All the dotted lines represent the absorption spectra, and all the solid lines represent the emission spectra. The figure includes the products within visible range. And the right top is the NIR1 product, and the right bottom is the spectra of NIR2 product. From all these spectra, we can know that AIE dyes provide a unique spectra with a broad emission and a large Stokes shift, meaning the absorption peak and the emission peak are far away from each other. so that AIE dyes are not prone to quench itself. In addition, we have also validated our product for 2-photon microscopy applications. And here is the example of a 2-photon absorption spectra of Luminicell Tracker 670. Comparing with another red emissive dye, Evans Blue, we can tell that Luminicell Tracker 670 has a much higher sensitivity for 2-photon excitation, which results in outstanding signal-to-noise ratio, and it further enables better depth of imaging. After introducing the products, let me share about the performance validation. Firstly, we validate the brightness of the products. Here we compare our products with quantum dots, as they are one of the brightest products in the market. From the two figures shown here, we can tell that each Luminicell tracker emits 40 times more photons than a quantum dot captured in 100 seconds, meaning we have ultra high brightness. Next, we tested the physical and the photo stability of Luminicell trackers. From the left, we know that Luminicell tracker has a higher physical stability than quantum dots, and the right figure tells us Luminicell tracker has a higher photo stability than FITC, which is a small molecular dye. After validating the physical data, we move on to test the cell labeling efficiency in different cell lines. As shown in the confocal images on the left, comparing with the gene transfection method, Luminicell Tracker is a non-transgenic labeling method, and it can achieve rapid labeling with high cell labeling rate, and it works well with different cell lines. Moving on, we also validated the capability of Luminicell Tracker for cell lineage tracing purpose. From the flow cytometry data and the confocal images, we can conclude that Luminicell Tracker can trace the cells in vitro for more than 10 cell generations. And comparing with quantum dots, Luminicell Tracker has a 3 times longer tracing time and 10 times higher brightness. Last but not least, we also validated biocompatibility of Luminicell trackers. We know that biocompatibility is rather important for biological applications. From the data on the left, we know that there is no significant body weight change of mice after treated with Luminicell trackers, and there is no lesions or damages caused to the organs. And therefore, there is negligible biointerference caused by Luminicell trackers. Also, we have validated the cell toxicity in vitro. After incubation with Luminicell trackers for 48 hours, the cell viability is still higher than 95%, indicating negligible cell toxicity. Here comes to the key advantages summary of Luminicell trackers. We have a 10 times brighter signal, and we have a 3 times signal longevity. We are suitable for long-term tracking, and we have good biocompatibility with negligible biointerference. In one sentence, Luminicell trackers are designed to be the best-in-class enabling solutions for end-to-end translational biomedical research. After sharing so much information on design principles and product performance validation, I guess you must be wondering what applications our products can be used for and what advances can be made in translational biomedical research with Luminicell trackers. Here comes the applications. We have categorized the applications into 3 different fields. And the first one is vascular imaging. This is the application for our vascular tracking dye. And we have the key benefits here, including the high brightness and outstanding photostability, and most importantly, the compatibility with multiphoton microscopy, which enables the intravitreal imaging and mapping of vasculature. So we can be used for the cases like brain vasculature imaging and also the vascular leakage detection. And another type of application is the cell-cell interaction study, and it is the application for the cell trackers. Beyond the above benefits, we also have the cytosolic labeling, rapid and efficient cell labeling, and good cell retention. And importantly, there is no crosstalk between different cells, meaning there is no leakage from the healthy cells. So the cellular trackers can be used to stain the immune cells and the cancer cells to study the immune cell reaction to cancer cells, which is meaningful for the immune cell biology and cancer cell biology study. And finally, we'll have the in vivo long-term cell tracking applications. The benefits adding here are the excellent biocompatibility and also the compatibility with in vivo imaging system. So Luminicell trackers can be used to stain the cells for cell therapy and to track the cell distribution post-transplantation, and also to track the cell viability and fate, which is a meaningful process for the cell therapy development. Now let me introduce these applications one by one. Firstly, how to map brain vasculature and how to detect vascular leakage. This page shows examples of vascular imaging with multiphoton microscope. On the left is the clear imaging and 3D reconstruction of mouse brain vasculature systems. And the brain vasculatures were labeled with Luminicell Tracker red and green respectively. The high resolution of the images can tell us the high brightness and the high signal-to-noise ratio. And on the right are the real-time intravital imaging of the bone marrow and eardermis vasculature tested with a 2-photon microscope. And here are the videos of each. We can observe the blood flow in the vessels after labeling. And Luminicell Tracker can also be used for monitoring of vascular integrity. We know that malaria infection causes hemorrhage, and we want to detect the blood hemorrhage site. So in this study, Luminicell Tracker, vascular tracker, has been applied to stain the blood vessels of the uninfected mice and also the infected mice. And Evans blue, another red emissive vascular dye, has been used as control. From the left images, we can see that uninfected mice have intact vasculature, and infected mice have blood leakage, and Luminicell Tracker can identify the leaking size. On the right, we cannot observe any significant difference between the healthy and infected mice. In conclusion, Luminicell Tracker can monitor the integrity of blood vessels and can be used for early identification of hemorrhage sites when there is infection or inflammation occurs. And here comes the cell-related applications. Firstly, how to study the cell-cell interaction using Luminicell trackers. Because we know there is no leakage from healthy cells, so Luminicell trackers can be used for cell-cell interaction study. In this study, Luminicell Tracker stained the triple-negative breast cancer cells MDA-MB157 to red, and the adiponectin-expressing Treg cells to green. And from the fluorescence images on the left, the red cancer cells engulfed the green Treg cells, showing a cell-in-cell phenomenon. And also the cancer cells exhibited the cell death after engulfment. This staining technology could be meaningful for studying how immune cells react to cancer cells and how they function. Here is another in vitro application to monitor the 3D cell culture with bottom-up staining. The 3D cell culture, including the organoids, spheroid, and tissue constructs, is a topic becoming more and more popular. And this topic is still under development. And we want to monitor the formation of the 3D culture. Here is the solution we proposed, bottom-up staining method. This project is collaborated with our German partners, Cantor Lab and Perspective Instruments, which is a multiphoton microscope company. Briefly, in this project, Luminicell Tracker has been used to stain the cells before forming spheroid, and afterwards, the whole process of spheroid formation is monitored under microscope. The beautiful image shown on the left is the spheroid harvested on day 7 after formation, and it got fixed and also stained with Holchast and Philodin post-fixation. And the video on the right shows the layer-by-layer scanning result of the spheroid, from which we can observe the strong red signal emitted by Luminicell from every layer, proving the brightness and longevity of Luminicell trackers. If you are doing 3D cell culture or plan to do so, I hope our products can provide you some new ideas. Last but not least, Luminicell trackers can also be utilized to track the in vivo distribution of the transplanted cells. The first application case study is the long-term tracking of tumor cells and also the monitoring of tumor formation process. In this project, Luminicell Tracker 810, which is a near IR1 emissive product, has been used to stain the tumor cells in vitro, and the stained tumor cells were subcutaneously transplanted into the mice. Q-Tracker 800 was also used as a control for the whole experiment. From the IVIS images on the left, we can tell that after day 31 of transplantation, the signal of Luminicell Tracker can still be clearly detected, while for Q-Tracker labeled cells, the signal can only be seen on day 5 or day 7. And the red quantitative data figure also tells us that the Luminicell Tracker 810 has a much higher brightness and a much longer signal longevity than quantum dot tracker 800. Our method is non-transgenic, meaning we're compatible with primary cells, and we're compatible with patient-derived tumor cells. So we can be used to study the patient-derived tumor models, which is meaningful for the drug screening purpose. Here is another application case study, long-term tracking of adipose tissue-derived stem cells. In this study, Luminicell Tracker has been used to stain the adipose tissue-derived stem cells both in vitro and in vivo. From the confocal data in vitro shown on the left, we can observe that Luminicell Trackers has a much higher brightness and a much longer signal longevity than the controls PKH26 and Q-Tracker 655. And the in vivo data also tell us that after stained with Luminicell Tracker and transplanted in vivo, the signal of the stem cells can still be detected on day 42 post-transplantation, also proving the brightness and longevity of the Luminicell Trackers. And here is another case study using Luminicell Tracker to track the stem cell-derived neurons in vivo. Firstly, Luminicell Tracker has been proven with negligible toxicity caused to the stem cell-derived neurons. And for in vivo application, Luminicell Tracker stained neurons can still be detected even after 1 month, shown on the right images. And without the benefits we concluded here, the long-term tracking could not be achieved. And here are examples for the in vivo tracking of xenogenic cells for immunotherapy. So in these 2 studies, xenogenic cells have been applied to activate the immune system of the tumor-bearing mice to induce immunotherapy. Briefly, xenogenic cells were stained with Luminicell Trackers and injected into the tumor site in situ. From the images, we can observe the signal of Luminicell Trackers until day 21. In addition, the researchers also applied PCR to track the location and longevity of the xenogenic cells post-transplantation. And Luminicell signals are well correlated with PCR data, proving the capability of Luminicell Trackers for long-term in vivo biodistribution study. Here are 2 more case studies. On the left, Luminicell Tracker was used to stain and track the biodistribution of the MSCs, and the organs were harvested after 2 weeks of transplantation. And from those ex vivo organ images, The transplanted MSCs have a significantly higher signal in the lungs, and indicating that the MSCs were homing and accumulated in lungs. On the right, the researchers make use of the long-term tracking ability of Luminicell Tracker to scan MSCs and study the different effect of a reduced and oxidized HMGB1 on the tumor infiltration behavior of the MSCs. And these data are as the examples of our proven use cases, but we're not limited to these cases. Here is the summary of the benefits of Luminicell trackers. We have a high brightness, which enables researchers to visualize with a high signal-to-noise ratio and lower detection limit. And we have a signal longevity, opens possibility to perform long-term live-cell imaging. We are highly biocompatible, compatible for in vivo applications. We are non-transgenic, meaning there is no genetic modification involved, not affecting the cell's normal behavior. According to different users, our protocol is easy to use. And more importantly, we're customer-centric. We have a technology that allows us for faster turnaround, and we're more than happy to provide customized solutions to accelerate translational research. After listening all the above, I hope you are still interested to learn more about Luminicell trackers. And here's our official website. You can learn more about the product information, find our technical notes, and also purchase Luminicell trackers from our e-store. Also, we'd like to invite you to follow us on LinkedIn channel to read our weekly sharing regarding the fluorescent imaging topic and to see our updates of events, and also to catch our limited promotions. That's almost the end of the sharing. Please reach out to us if you have any queries regarding product information or applications. I'd like to invite you to scan the QR code and help us complete the product interest survey to let us know your interest and needs so that we can support you better. Thank you for listening.